Okay, sorry, everybody. Okay, so now let's try to avoid the echo or whatever it is. Okay, so this was obviously, for those who are my age, a kind of reference to Woody Allen's movie from the 70s. And I hope that I can make it, I know that for most of you, anesthetics are a completely kind of uncharted territory, but I hope I can make it interesting enough for you and that you will be able to follow me. So let's now see that the things work. Okay. Well, that doesn't work. Okay, fine. Huh. Where is our IT person? So it doesn't work. Ah, here we go. Hang on, slideshow, resume slideshow. Let's try this one. So I wanted to start off by saying that I'm really glad. Oh, here we go. So I would like to start with the key points. And as you can see, and David mentioned, general aesthetics are highly promiscuous drugs. And there are two kind of big aspects to it. One is the visible aspect. This is what you all think about when you hear anesthesia. But that's only what is, I'm using the iceberg analogy, what's above the surface. And then below the surface, there is a huge unknown territory of unknown collateral effects or off-target effects. And we have two models to investigate these collateral effects. One is the closed-set TBI that was developed by David and Barry. And the other one is the mitochondrial mutant, which is also the first experiment was done thanks to Karin Löwen and Barry Ganetsky, characterizing a specific mutant. So I will be bringing an example on the closed-head injury, but mostly I will be talking about the mitochondrial mutants. But because of number one, so that these drugs are so non-selective. Basically, I came to the conclusion that genetic approaches are probably the best way to decipher or to untangle all the things that happen. So it doesn't go up here.
Advancing on Zoom.
Yes, I don't know what to do about that. I mean, this is just, so how do we advance? Or maybe if I click this, no, Zoom doesn't advance. It advances over there. Yeah, I'm sorry about that, but so how are we gonna, so I'm clicking forward. I kind of have to click resume slideshow every time. Okay, so I will continue talking about here. I'm sorry for everybody who's on.
Yeah, I'm gonna change the screen sharing mode.
Okay, so this, okay, great, good idea. Let's see that it works. Okay, so I mentioned that, in my opinion, the genetic strategies are the way to understand what molecular actors are participating in this whole thing. Good, now it seems to be working. I have it advanced. And so from the beginning, there were three core questions of anesthetic pharmacology, and I'm going to touch on them first. Then I will discuss our central hypothesis, the hypotheses that are driving our research, have been driving basically from the beginning. And then I will introduce the mitochondrial mutant as our model and what we can learn from it. So the take-home message is that pharmacogenetics play, in my opinion, an important role in potential adverse effects surrounding the perioperative experience, and that deciphering them will be a step to personalized perioperative medicine. So you can see this quote here on the side. You can read it yourself. I don't have to talk about it. So anesthetic pharmacology, the question of how do anesthetics work? What is the mechanism of anesthetic effect has occupied very smart people since the early second half of the last but one century. A number of Nobel, starting with Claude Bernard, he was followed by a number of Nobel Prize laureates, including Francis Crick and others, who tried to crack and come up with a simple explanation of how anesthetics work. So then the other big question of anesthetic pharmacology are the conventional complications. And this is mostly what physicians have been dealing with for the last 50 or 60 years, how to recognize them, how to treat them, and how to possibly predict them. And the third is this collateral effects, which is going to be the center of my topic. And they have been actually, although the notion that something can happen has existed for a long time, but they have really gone into the center of our attention probably only in the mid to late 90s. And since then, it's one of the big topics of research into anesthetic pharmacology. So as you can see from here, it's kind of not easy to make sense of anesthetics. And the reason is that you can see all these drugs with all these different structures, they all cause anesthesia. And so you can imagine that figuring out some kind of a receptor lock mechanism is kind of challenging, let's put it this way, right? And I have here put in square brackets the blue drugs. These are midazolam and propofol, which are the most widely used drugs in the operating room probably. And any one of you who has had some kind of a procedure under, even not necessarily general, but under surgery, you probably were exposed to either both of them or to at least one of them. Now, if you went on to have like real surgery in the United States, you most probably have been exposed to one of those drugs, either to sevoflurane or to isoflurane, which are both halogenated fluorinated ethers. So these drugs, we all get exposed to them. And so I think it's interesting to find out a little bit more about them. And so because of this diversity of structures, one of the first sentences that many of you will face in pharmacology textbooks is a quote from Hans Horst Meyer from the Meyer-Overton rule. You may have heard about it. It was formulated in around 1900. And he said that any otherwise inert lipid-soluble substance is an anesthetic. And this was a dictum. And so this, well, not only this, but the history of research into anesthetics was shaped for more than a century, for probably 120 years, by two correlations, right? And it's important to remember that correlation is not causation. But there were two influential correlations. One shows the correlation between lipid solubility and potency. So you have lipid solubility with lambda oil gas partition coefficient on this side, and you can see this ranges from like over a 10-fold logarithmic range, and the potency expressed as MAC, but it's not important what that means. But you see that there is an incredible correlation between lipid solubility and anesthetic potency. And the other correlation that began interesting or got on the stage in the 1990s was also an amazing correlation, also across a wide range of concentrations and potencies. And this is the correlation between anesthetic potency and effect on an enzyme, on a purified enzyme. This is important because while a lot of people have thought about proteins as being the target of anesthetics decades and decades before that, but here it was a crystallized purified protein, and Nick Franks and Believe could show that these two correlate also. So where does it leave us then? And so the whole thing prompted or was guided by the notion that there must be a unified hypothesis to explain anesthesia. All these different drugs, lipid is a target, linear correlation, there must be a single, and you can see how Thomas Butler 70 years ago formulated this, that this search for a single theory and this great desire to unify may have delayed arrival at a more satisfactory solution, which is actually the case. So one of the solutions for that was to break up. I'm sorry, I still have this live transcription channel. Okay, let's close that. Okay, so now the researchers who investigate mechanisms of anesthesia, they have broken this unified state into three substates: amnesia, hypnosis, and mobility, and try to investigate them separately. And you can see by this very simple schematic that the hypothesis is that they have different dose response curves and that they also act at different parts in the central nervous system. And either or. Okay, I'm stuck again here. Let me see what's going on. So is our IT person gone? Okay. So well, I'm sharing the screen. The problem, I cannot advance it That's the problem. We had it at the beginning, and now it's like stuck. Oh no, here it's now it's like this. Okay, good. And so basically, amnesia and hypnosis are necessary to suppress the experience of surgical experience. The immobility is largely a matter of convenience for the surgeons and for us as well. So they're being investigated separately. Now, the second question. We talked about the mechanisms of action of general anesthesia, and now the second question is the conventional side effects. What do I mean by conventional side effects? They are detectable with conventional monitoring. This is what we look for in the operating room every day, and this is basically what you pay us for, because our drugs are pretty good to cause anesthesia, but it's the side effects that you need a professional to deal with them. And so there are those which are very frequent, postoperative nausea and vomiting and pain, and you may have experienced them. Then there are those which are predictable, treatable, and avoidable to some degree, mostly cardio and cerebrovascular complications. And then there is this big and vague complex of postoperative neurocognitive disorders. And they occur on different time scales. They can be either immediate, short, or very much delayed and long term. And so now we're switching to the third question. And these are the unconventional or off-target effects. So first of all, one characteristic thing is that they are unpredictable. However, it is important to understand that they are not allergic in the medical sense or in the physiological sense, because they're not mediated by any kind of allergies. The most important one is malignant hypothermic syndrome. It has a very dramatic phenotype. And what I call it is like you have the wrong S&P, and then a completely safe drug becomes highly deleterious for you. So then there are all those which are not detectable with conventional monitoring. And one of them is, for example, this big question of immune modulation. Do anesthetics affect the immune system in any kind of physiologically significant way? And I think that we don't know what the answer to this question is. Then there is this complex of anesthetic-induced neurotoxicity. And this is kind of, we're getting close to what we're investigating in our group. And you have them in the immature brain. This is where this term has been coined about 20 years ago. And the postulate is that they have long exposure to anesthetics of the developing brain have long-term consequences. Now, I somewhat flippantly call it lab evidence in search of clinical relevance, because there's really no clinically relevance, clinically documented, or shall I say, phenotype, but other people would argue with me. And then there is the other one, which is in the aged brain, and this is what is referred to with perioperative neurocognitive disorders, which is definitely there. I mean, this happens certainly, especially on the short time scale of hours, days, and maybe weeks, but there is really no laboratory model which would reproduce it. And this is why I call it, like we're looking for laboratory evidence. And then there is the third thing, which I haven't seen it in the literature, but I would call it the vulnerable brain. So you may have a brain that has been sensitized to these collateral effects of anesthetics. And the situations that I can imagine are specific single nucleotide polymorphisms in some proteins with which anesthetics can interact. Anesthetics interact with almost every protein, as long as it has a cavity of more than 100 angstrom, cubic angstrom. So, and there are lots of them. So then TBI, traumatic brain injury, and I will show you an example. Traumatic brain injury also does something to the brain, which changes the brain's interaction with anesthetics. And then there are probably also other neurodegenerative disorders, but I haven't studied them under condition, and other people have not studied them under conditions which would really be capable of isolating the effects of anesthetics. And so if I summarize it, this iceberg of anesthetic effects, there are the main effects, behavioral, there are the side effects, mostly observable and in the cardiovascular system and the central nervous system. Then you have those collateral effects, some of them well documented, other more hypothetical. So now part 2 of my talk, and that is central hypothesis. The first hypothesis is that the significance of anesthetic interactions is biological context dependent. It means that what the anesthetic are going to do to whatever molecules they interact with, and how this is going to be reflected in some kind of phenotype, will depend on the state of the biological system at the time when the interaction between anesthetic and the brain takes place. And for this, in order to study this, the flies are probably perfect system, but we have to somehow get the anesthetics to them, right? So we have to devise a system to administer both anesthetics to flies. And this system, what we wanted to have is something simple, reliable, with high throughput, and very importantly, that we could simultaneously test multiple experimental groups under exact same conditions. Because if we're going to go into the details, we want to be confident that what we're doing is exactly the same to the different groups that we're going to be comparing. And for this, and this is how the serial anesthesia array was born. This is how it looks. And this is who built the first one, Model T. This is this Model T, Mark Perkins from Bob Pierce's lab. And so what it consists of is an air cylinder, or it could be also oxygen or whatever other gas, which gets the gas to the flow meters. In the flow meters, you set the fresh gas flow at what rate the gas is going to flow into the vaporizer. The vaporizer, this is a clinically active vaporizer that I managed to get from the hospital. In the vaporizer, the gas mixture gets mixed with a certain precisely set percentage concentration of an anesthetic, and then it goes through this system. And we send it first through a water bath to try to humidify it, because we didn't really know how long the exposures may be, and we didn't want the flies to dry out. And then you have 8, in this construction, you have 8 slots where you can put different experimental groups or the same experimental groups with for replicates. And then at the end, and I cannot see from where I'm standing here, but there is a flow meter. This is like a safety thing, because you want to make sure that the stuff is really flowing to them, because who knows? I mean, there can be some obstruction or something like this. And here is, yes, it is the graphic. So this is a large version of the system. So the flies at the bottom, after they have been anesthetized, inflow and outflow from every single one of those slots. And so now I'm going to show you the SAA in action. And I'm going to tell you, because it's going to be fast, what you will see. So these hands here, I don't remember whose they are. Zach is going to connect the first vial to the system, and then gas will flow through the system. And what you will see is a white cloud that's going to fill up these slots, and you will see that this thing starts, oops, sorry, no, this was the wrong, this thing is going to start rotating to showing that there is gas flow. So let's see, yes, it works. Okay, so he screws down. You see this gas is flowing, this thing rotates, and then you can see how this white cloud propagates from cylinder to cylinder. So it's the system, and we've tested this. I mean, it's fast, it's reliable, it's exactly the same concentration in all those locations, so we are pretty confident that it works. And now I'm going to show you what happens when you expose flies to the drugs. And here it's also going to be fast. You see the flies, these little black dots are running around. Anesthetic agent is coming in. Do you see anything moving there? So you see they're climbing around, and then they will start getting immobilized. And this is basically fundamentally what happens with anesthetic. Okay, so we have a system where we can administer precise concentrations and precise time, precise doses of anesthetics. So, and now I will show you the kind of the meat of the first series of experiments, and that is, let's just get used to seeing these graphs. So on the y-axis, we have an index of mortality, simplified in percent. And this is the mortality 24 hours after a traumatic brain injury. And I'm just illustrating this as an example. I'm not going to be talking about it. And now if you expose the flies before you give them the traumatic brain injury to isoflurane at a certain dose, you will get a significant suppression of 24-hour mortality. Okay, this is a 50%. And I will show you now this thing worked up a little bit with more data. And you can see that exposure of flies prior to a traumatic brain injury to either isoflurane or sevoflurane reduces the mortality by approximately 50%. Now, the interesting thing is, and the biological context comes into play, that if you do this after TBI, the same drug, the same dose, the same flies, that you have with isoflurane, you increase mortality. So you see a toxic effect of isoflurane exposure, while sevoflurane has no effect. So this is kind of the meat of the experiment. And this is experiments were done by Julie Fisher. She's a genetics graduate here at UW. And later on, it was developed and done more experiments by Hannah Schiffman. Okay.
How long before you give them TBI? How long before?
Well, the typical experiment is we started off with no interval. So but then we went to 5 minutes interval. And we know that with isoflurane, you can extend the interval to up to 60 minutes. You will still see an effect. However, by 90 minutes, you don't anymore. So it's a short, well, it's not necessary. This is a very good question. We can talk about it afterwards, because whether it requires the presence of this anesthetic in the brain during the TBI, this is what you're kind of going to forward, or whether it's an effect that's set in motion, that's the question. And we have, as I said, we cannot exclude that there is still, after 1 hour, there is still some anesthetic, but the size of effect declines with time.
5 minutes pulse an hour before, or is it time?
No, it is. It is the experiments that I've shown just now is 2 hours of isoflurane, then 5 minutes interval, and then the TBI. We have shortened this exposure duration to 15 minutes, 1.5. It's the same size of effect. So this was kind of the proof of principle, that biological context does affect anesthetic pharmacodynamics. And now comes second hypothesis, that the genetic background influences anesthetic pharmacodynamics in Drosophila. And so I will show you another movie here. It was the same director who made the movie, Zach. And so you will see on the left side, we will expose the flies a control, which is white 1118 for those who are in the fly world, and a mutant. And we're going to now flush the anesthetic out. So we will see how they recover from anesthesia. And in order to make it a little bit, so you can see that here the flies are starting to move with anesthetic number 2. The mutants are starting to move. Well, here everything is flat out. And I will then tell you why this is important. You see how these guys run around. This just start, and there is complete silence, or basically complete silence under anesthetic number 1. Okay, I hope I have convinced you. So now I will tell you 2 things about. First of all, we're going to show you some stats. But the important thing is that the anesthetic effects in flies reproduce what we see in humans, and that is that one drug, the yellow drug, sevoflurane, is less potent than the purple drug, isoflurane. And this is here expressed with the EC50. We use a behavioral assay to quantify the effect of anesthetics. You can see that EC50 for isoflurane is about half of what it is for sevoflurane. It just so happens that this is approximately the same correlation as we have in higher animals and vertebrates. So this was reassuring that we reproduce this specific effect in 2 drugs. Now, emergence from anesthesia. And this is what you saw in the last movie. How quickly do the flies start moving again? And here again, we see exactly kind of what we would expect from mammalian models. And that is that it takes longer for isoflurane to regain mobility after isoflurane anesthetics than it takes after sevoflurane anesthetics. So here again, we reproduce the relative kinetics that we are familiar with in mammals. And now, the third thing was anesthetic potency increases with age, which means that with age, the EC50 decreases. And here again, you can see that for both drugs, as we age the flies, the EC50 decreases. Again, these 2 drugs we're studying. And so, yes.
Did you look for hysteresis?
No, because we don't have the time resolution and the behavioral assay to look at induction. Now we could, well, I know we don't really have to, we would have to, let's put it this way, we thought about it, we didn't do it. Okay. Because we're not interested in the mechanisms of action of anesthetics. This is just to make the proof of concept that these flies in our hands reproduce what we typically assume as being a given for the classic drugs. So, and now the genetic background question, because we're getting like really close to the meat of what flies can provide you. And so an essential tool that we have as Drosophila researchers is available to us are the collections of inbred fly lines. And there are two big collections. One is called the DSPR, 8 founder lines, 1,600 recombinant inbred fly lines. And the other one is the Drosophila genetic reference panel. And this is the one that we're going to be showing you results from. The founder flies were caught out of the wild in around Raleigh, North Carolina. And this is why they are identified with the prefix RAL. And by now there are over 200 inbred and fully sequenced fly lines and over 4.5 million SNPs identified. Now, the important thing is that this is caught from the wild. So these are variations in the genetic background, which are available in nature. So that we don't, they are not mutants. They just have it. And so now let's have a look at what we found with them. So we subjected, here are the different RAL lines. The white 1118 serves as control, purple isoflurane, yellow sevoflurane. And you can see that the EC50 varies between the different lines, right? Both for isoflurane and for sevoflurane. And also the time to recovery, that's the speed at which they regain mobility, also varies between the different RAL lines. Now, importantly, it's always, for the same RAL line, it's always isoflurane more potent than sevoflurane, and sevoflurane faster than isoflurane, which is very reassuring because I would have a hard time to think about how we would explain it if it were not this case. And so here we plot the EC50s one against the other, and if they were in a completely the same relationship, we would have a straight line through here and the same thing over here, but you can see that they're kind of scattered around a little bit. So they don't go hand in hand. While sevoflurane is always less potent, but it's not always the same proportion between those two. And so this was the proof of principle number 2, right? Genetic background does modulate pharmacodynamics. And now we're going to switch gears and we're going to go to the model itself, the mitochondrial mutant. And I will first give you a quick overview over the electron transport chain, because this is what we're going to be talking about. So as you all know, the electron transport chain generates electrons and protons, and then finally they're going to generate the ATP. It consists of 5 complexes. Complex number 1 is the biggest of all of them. It consists of about 46 subunits. And it has these 2 domains, one which is in the membrane, it's called the intramembranous domain, and the other one, which is the domain in the matrix, which protrudes into the matrix of the mitochondria. And then you have the complex 2, complex 3, complex 4, and complex 5. Complex 5 is ATP synthase. And they move electrons, they establish a electrochemical gradient across the inner mitochondrial membrane, which then serves as a pump to drive ATP synthesis. And in the process, one of the many known, or I'll show you the best known byproduct, which has an amazing complicated role in cell physiology, are reactive oxygen species. So note that they are here illustrated in different colors because we're going to come back to that. And so if you think back about the EC50s, there was one outlier, which was more sensitive both to sevoflurane and to isoflurane, and substantially more sensitive than all the other RAL lines. And this one is the mitochondrial mutant. It's a mutation in the ND23 subunit of complex 1. I show it here. This is the location of ND23, and you can see that it's in the matrix domain of complex 1. And so it was characterized, this specific allele was characterized by Karin Löwen and Bereganetsky. And one of the important aspects of it is it is nuclearly encoded. And I will come back why this is important. And it's autologous to Endus 8. And both ND23 and endosate, mammalian endosate, have been proposed as being models of Lee syndrome, which is a severe neurodegenerative disease carried or affecting patients carrying mutations in complex 1. Now, the question was, after having a great different models, why deviate, why change course and go towards mitochondria? So anesthetics have also been in some way or other implicated into some interaction with mitochondria. Already in the 70s, it was shown that anesthetics depress cellular respiration. But the really big thing for me happened in the 1980s when Phil Morgan and Mark Sedensky, and here they are shown attending Woodstock, I think, they did an EMS screen of C. elegans. And they were looking for mutants with changing in life expectancy, alcohol sensitivity, and other stuff. But they identified one mutant. They're both anesthesiologists, pediatric anesthesiologists right now in Seattle. So they identified one mutant, which was originally called FC21, but then they changed the name to GAS1, which is general anesthetic sensitive. And this was the one mutant that was sensitive to all multi-general anesthetics that they have tested. This is why GAS1. And so what they did then, they identified, sequenced, and invested a lot of effort over 10, 15 years. And finally, in 1999, they found that gas was actually homologous to the 49 kilodalton subunit of complex 1, which is like a giant subunit inside complex 1 itself. And then once Phil Morgan started kind of looking at it and got really interested, he published a case series of children, which all had some kind of mitochondrial mutation. They are all very symptomatic. Most of them are very sick. And most of them come for a muscle biopsy in order to make a genetic diagnosis of what they have. And only the kids that had a mutation in complex 1 were hypersensitive to anesthetics. And so from this, we knew, or the conclusion was that complex 1 is a behaviorally relevant target for the behavior, because we have a change in behavior in mutants. And so the question that we decided to ask was, are mitochondria also linked to the collateral effects of anesthetics, to anesthetic-induced neurotoxicity and or perioperative, post-operative neurocognitive disorder? So that's kind of like, we come to the meat of the project. Now, this is a very complicated slide, but it's relatively easy. You recognize this part. And the reason why I'm showing it is that it's beautifully color-coded what is coded in the nuclear DNA and what's coded in the mitochondrial DNA. And you can see that basically all the subunits, 1, 2, 3, 4, cytochrome C, and 4 and 5, they all are, some subunits of them are coded in the nuclear DNA. And there are fewer of them, only 1, 2, 3, 1, 3, 4, and 5, which also have subunits from the mitochondrial DNA, right? So which means that, for example, complex 2, succinate dehydrogenase, is completely coded in the nucleus. And that's important because it first of all generates a very complex inheritance pattern for mitochondria. It took a long time to start understanding what's going on there. 80% of the diseases are caused by mutations in the nuclear DNA. And typically, they manifest themselves already in childhood. Why? Because they have to be homozygous in order to manifest themselves. So this mutation, the typical mutation in the nuclear DNA, is recessive, mutation affecting the proteins in the mitochondrial electron transfer chain. Now, there are also mutations in the mitochondrial DNA, but these typically manifest themselves only in the adult at different ages, anywhere between 40 and 60. And the thought is that because of all this Ross generation in the mitochondria, these mitochondria accumulate damage over your lifetime. And the more damage, the more prominent even a small defect in the protein is going to be manifest. It's also possible that mutated proteins are more susceptible to accumulating damage from Ross. But for all these rules, there are many exceptions. Okay, so when did the era of mitochondrial diseases begin? It's actually not that long ago. In 1988, the first mitochondrial DNA mutation was linked to a clinical disease, mitochondrial DNA, right? It was done by Doug Wallace, and the diseases, they published 2 papers in the same year. One was Leber's hereditary optic neuropathy, which with advancing age manifests itself with, well, what this is, an optic neuropathy, you get blind. And the other one was a neuromuscular disorder, which manifested as in adults, and both of them were caused by mitochondrial DNA mutations. Now, since then, this field has exploded. There are hundreds, thousands of papers on this topic. And so by 2010, also nuclear DNA mutations were identified that cause a phenotype. And this is typically referred to as the Leigh syndrome, which is a very severe, lethal neurodegenerative disorder. And in 2010, a mitochondrial mutation, NDUFS2, was linked also to Leigh syndrome. So here, mitochondrial DNA, and the other one's nuclear DNA. And then we have, in parallel, we're developing mouse models. So the first was the NDUFS4 mutant. Now we're talking about mutations affecting the mitochondrial electron transport chain. And the mitochondria have way more proteins than just those which are in the mitochondrial electron transport chain, but we're talking about that chain. And then in 2014, the ND2, the nuclear mutations, was released as a Leigh syndrome model. And then this was followed in 2018 by the model that Barry and Karen created here in Madison. So now we're going back to the collateral effects. So we have Leigh syndrome. It's a very severe disease. Babies, when they are born, the suspicion comes out with the pediatrician immediately. They are referred to as floppy babies. It's incurable, right? They are doomed to die within 2, 3, 4, 5 years. And they are very rare. Live births, homozygous, are 1 in 20 to 40,000 cases, except for hotspots. There's one in Canada, there is one in the Faroe Islands, where the incidence of live births is about 1 in 5 to 1 in 7,000. And so there are mammalian models. And you ask yourself, why bother then with Drosophila if you already have mammalian models? And there are a couple of reasons for it. First of all, if you remember the Hardy-Weinberg equilibrium, if you have an incidence of homozygous carriers of 1 in 20 to 40,000, the incidence of actual carriers, heterozygotes, is much, much higher. And for these diseases, for the complex of Leigh syndrome, it's probably between 0.5 and 1% of carriers. We don't have 100 people here, but if we include the Zoom people, so there will be 1 or 2 of you who do carry a mutation which, if homozygous, will present as Leigh syndrome. Now, however, what is interesting that the carriers are not diagnosable. They don't have a phenotype in normal life. If you're heterozygous for a nuclear mutation, your remaining gene, the one that is normal, covers it. It's haplosufficient. And the question is whether there are conditions where we suddenly can see a phenotype that comes from this recessive trait. And so we went on and started characterizing the homozygous mutations. So first of all, what we found is isoflurane is toxic ND23/6 in this mutation, but it's age dependent. This is days after hatching, and you can see that in the first couple of days, these animals don't die. They don't have a toxicity phenotype. They do have a behavioral phenotype already here, but they don't have a toxicity phenotype until at about 10 to 13 days of age. You have a substantial mortality from a 2-hour exposure to isoflurane recorded 24 hours later. So we wondered whether the toxicity is really dependent on the ND23 being mutated. And so Zach introduced a couple of alleles into the flies, and we can show here that if you mutate ND23, no matter which allele you use, you will have this toxicity. And then the interesting thing is also that it is dependent on both the agent and the oxygen concentration. So you have here 2 parameters, sort of 75% oxygen or 5% oxygen. These are recorded in normoxia, in 21% oxygen. here isoflurane, sevoflurane, 75% oxygen, 5% oxygen. And you can see that in 75% oxygen, hyperoxia dramatically increases mortality. Like you get 100% mortality in the homozygous slice with isoflurane, but not with sevoflurane, a very low mortality, if any, with sevoflurane. However, if you go down to 5% oxygen, hypoxia, you have a dramatic rescue effect. The mortality from isoflurane drops into the order of 10%. So that's a dramatic rescue effect. And this is also something that has been observed with chronic hypoxia and hyperoxia exposures in mouse models. But here, this is a 2-hour exposure, which results in a very strong phenotype. And so we also wondered whether the toxicity that we observed with ND23 mutants is due to its action in the brain. And so what Zach did here, he overexpressed wild type ND23. He overexpressed it ubiquitously, and you get a complete rescue. He overexpressed it in Europe. So the rescue is here. So this is the mortality in the ND23 mutant, and this is the mortality with ubiquitous overexpression of ND23, it's 0. If you overexpress it only in neurons, you also have a basically complete recovery or rescue from mortality. So neurons must be heavily involved into this. If you overexpress it in glia, you get a substantial rescue of about, well, you see there is a big error bars, but of about 40 to 50%. Now this is remarkable because in Drosophila, only 10% of the cells in the brain are glial cells, as opposed to the mammalian or human central nervous system, where it is much higher. We have a little bit of an argument about exactly how much higher, but it is substantially higher. But here, even this little 50% of 10% of glial cells cause a dramatic rescue. And here is an even more dramatic rescue. This is the rescue when you overexpress wild type in dopaminergic neurons, and which are just a handful in the Drosophila brain. And you can see that here also you can rescue the flies. Unfortunately, so far we haven't been able to pursue this line. So the question is now, is it relevant beyond Leigh syndrome, right? Because I said it's quite rare. And this is where the Hardy-Weinberg equilibrium comes in, because we can show that under certain stress conditions, we can maybe reveal a phenotype in heterozygous. Now, what you've seen until now were homozygous mutants. So the homozygotes have a short lifespan, and they are vulnerable to anesthetic toxicity, as I have shown to you, in approximately midlife. And Karin and Barry have characterized the lifespan of these animals. So you can see here, it's a little bit more complicated than necessary. Here you have the canton S. Here you have the homozygote, ND23 mutant, lifespan 20 days. And here you have the heterozygote, which has a longer lifespan than the wild type, canton S, which is frequently observed. It's referred to as hybrid vigor. Right? And now, so the heterozygotes have a normal, if not long, lifespan. And now we will expose them across, as they age, to this combination of isoflurane and hyperoxia. And you can see that while 15 to 22 and up to about 30 days of age, there is no phenotype. Nothing happens to them. They're just happy campers. But then suddenly mortality increases. And we thought that this was a striking finding, because this tells us that asymptomatic carriers are at risk from certain, not further characterized complications when exposed to stress in this case being a combination of an anesthetic, isoflurane, and hyperoxia. By the way, 75% oxygen in the operating room is not the standard, but it's very frequent. And plus, this doesn't take into account the stress of surgery. We don't know what the surgery in any way plays into role, but this is just from the exposure to anesthetics. And so, based on this, we want to go forward. We want to see what's going to happen further on. And this is kind of like a schematic of our grant. So we want to investigate how these heterozygote mutations affect how generalizable this finding is, okay? So, and for this, we have to identify what are the pathways that are affected in the homozygotes. The homozygotes have a stronger phenotype, so it's kind of easier to study the homozygotes and then do targeted experiments in heterozygotes. And this is what we have done. We've done a RNA-seq at a certain time point. We have, with the help of Mark Veris and Derek Pellis, we have established a experimental setup in order to be able to block, to apply statistical blocking techniques. And we've done this in 10 to 13 day old heads only, separately for males and females. And this is just a color picture just to show what you get. You get the Venn diagrams from which you can, through many combinations, try to figure out what is kind of, what do the drugs have in common. And what we found is basically that those genes that are highlighted in orange or in dark orange, on my screen it looks like dark orange, here it looks like brown. So the dark brown and the lighter brown are genes which are highly overexpressed after exposure to anesthetics in the mutants. And they fall, most of them fall into the chaperone and chaperone pseudogenes categories. And so we found this is a molecular kind of evidence that Volt anesthetics just on their own cause a stress response without any surgery. And so the two groups that we have investigated with RT-PCR are the stress response, oxidative stress response and chaperones. And we've done many more, but for the sake of simplicity, I'm just showing a couple of examples. So the conditions are hyperoxia, isoflurane, sevoflurane, and hypoxia, isoflurane, sevoflurane. And what you can see is that under the conditions of both hyperoxia and hypoxia, and this is kind of like one of the things that we don't understand, why we don't see a signal from oxygen. The phenotype from oxygen is obvious, oxygen kills. But on the gene expression level, what we see is the effect of both anesthetics. Now they're quantitatively different. Typically, for example, in the GSPD2, which is a oxidative stress response gene, we see a stronger effect with sevoflurane than with isoflurane, which we interpret that isoflurane suppresses. There is an oxidative stress, but isoflurane, for whatever reasons, suppresses the expression, while sevoflurane allows the expression. And you see the same thing in 75 and 5% oxygen. And then you have the HSP27, which is a chaperone. And here you see the opposite. You can see that isoflurane increases the expression of HSP27 way more than sevoflurane does. And we interpret this that it exerts a stronger stress, and that this overexpression is an adaptive response to some kind of a stress that is exerted on the cells. And so where we are. So we've asked relevant questions in a way that allows an answer, which I think that is impossible. These questions, asking these questions in mammalian models is basically impossible for a variety of reasons. And so we think that flies are genetic models that are useful tools to study the perioperative toxic effects of anesthetics and their effects in TBI. I've shown you the slide at the beginning. So currently, Amanda is studying the mitochondrial ND2 mutant. So the ND2 mutant is a mutant in the membranous part of complex 1. It's also in complex 1. It also is known to, in mice, to result in a Lee syndrome model, but we use it. First, it was a proof of principle that a different mutation in complex 1 will also result in a phenotype. And then being a mitochondrial mutation, it's relatively easy to introduce into other genetic backgrounds because it goes with the female, right? And so what we plan in the short term, we would like to figure out what the genetic signature for the toxicity is. We still don't know. We don't have actually, to be honest, we don't even have a clue. And then we will try to see whether mutations in other complexes of the electron transport chain result in a phenotype under the stresses of aging and exposure to anesthetics. This is not known, even though I said in the beginning that only complex 1 has a clear phenotype, but this is in kids and in C. elegans. But they've never looked what happens with aging, or when you expose them to a hyperoxic atmosphere, which is kind of like the standard treatment that you get in the operating room. And then we have a couple of course long-term goals, which are to identify the pathways which result in genetic risk factors for anesthetic toxicity in the wider sense. And we hope that this is going to be a step towards a personalized perioperative medicine. And so it's, of course, it's a team effort. And I've circled this part here is David Wasserman's lab, and I'm going not to identify each person, but those circled blue, Amanda, Zach, and just recently joining us, Luke, are working on the mitochondrial project. And Dina, who has joined us in January, is working on the TBI project, which keeps on going in the background. And I'm very grateful to these people who have, without whom it would have been impossible to get anywhere. Barry Ganeski and Karen Leuven, Grace, who participates in our journal club and gives always great suggestions about what kind of experiments to do. Obviously the Wasserman Collective. I'm supported by the Department of Anesthesiology for the TBI project and by an NIH grant where I work together with David for the mitochondrial mutant project. And that's the end of my talk. Thank you very much for your attention. Okay, well, it's exactly 1 hour, but I hope that somebody is willing to hang out and ask questions. Yes. It's a loss of function mutation, but it's not a complete absence. So you can still detect a residual protein of it, but it's smaller. It doesn't travel at the same speed. So it's not a complete knockout. It's a loss of function. Oh yes, Bob.
Very, very nice talk. This is going to filter into the clinician's consciousness. It's going to take some. A lot of the things that you talked about remind me of the phenomenon in anesthetic preconditioning. You didn't talk about it, others might know. Also, the anesthesia, sometime later, the organism is able to withstand hypoxia.
This is correct. We're studying, the reason why we're studying anesthetics in the context of traumatic brain injury is to find out what the pathways of preconditioning are. So preconditioning is a phenomenon from that was discovered in the 1990s. The first model was a short bout of hypoxia can protect excitable organs. It was done heart and brain from a subsequent injurious bout of hypoxia. And then somebody came around and discovered that actually anesthetics do the same thing. They don't use, they are very similar in effectiveness. They don't use exactly the same pathways. There are certain differences. And this was a big theme in the 1990s, but then it kind of got forgotten or pushed aside. I mean, it's for a variety of reasons. The main problem is that nobody used it in a system where you could actually go thoroughly and convincingly for the pathways. And I think that the goal is in this other project, the TBI project and preconditioning project, to do just that in fruit flies.
All right, so we're going to get back to the end of the question. And that was that you are showing some toxicities of the anesthetics themselves directly. And I wonder if just like hypoxia protects from future hypoxia, whether a brief bout of anesthesia protects from a later bout of anesthesia, so you'll bring right back into this part.
Oh, well, if I had known that you're going to, well, if I had known that you will be here, I could have expected that you would have asked the question. This is what Zach has been doing for the last, 2 months or something like this. We've tried all sorts of, in the quest to find out the pathway, we've tried different ways of preconditioning. And we haven't, including anesthetic pre-exposure. And we haven't found a single combination or modality that would protect from anesthetic toxicity. And it's quite mysterious. We're not quite done yet. We've tried heat shock. We've tried sevoflurane preconditioning to isoflurane, isoflurane to S1, all these combinations, different exposures. We never found the path, but we did find, and you correct me if I'm wrong, that if we administer both of these drugs together, the innocuous sevoflurane combined with the noxious isoflurane, the mortality goes up. And actually also preconditioning, right? Also preconditioning with isoflurane prior to isoflurane and sevoflurane increases the mortality. So it's the exact opposite of what we thought. We've tried to preconditioning with hypoxia, increases mortality. So there is still a lot to discover to find out how anesthetics exert the protective effect in this in our model, in the TBI model. Or actually, we've done this preconditioning stuff because we have such a strong phenotype. We've done it with the ND23s, right? So we will step and do it in the TBI model as well, but in the mitochondrial mutant, we couldn't find a path. And one of the possibilities is that by the nature of being mitochondrial mutants, they are incapable of setting in motion those pathways which are necessary to effect preconditioning, which would be also a useful result, because then we know exactly, well, not exactly, but then we kind of know where to look for in the TBI model. Yes?
This is a really question, but I couldn't help thinking about it. So Audrey Gash's lab has a environmental stress response that also preconditions survival. So it's in yeast. I mean, in yeast. And you probably know about this. So
No, I don't actually.
Yeah,
but I'm not surprised that it works in yeast. But...
Exposing to environmental stress first, later on, a better response, a better survival response to a subsequent stress.
Yeah. Well, this is yeast have nuclei, and I remember there was a great talk in this room, probably like 10 years ago, where Susan, forgot her last name, highly decorated scientist, she talked about this preconditioning effect, or not actually, not anesthetic, but stress preconditioning to other stress. And she brought the interesting kind of, how shall I say, an example. She said that it's kind of a Lamarckian effect. So we completely forgot, and actually it goes through generations. So you can stress the yeast, and their offspring will be, we will have some resistance to the stress. So it's something that gets inherited. And she brought this, that maybe Lamarck had something to what he was saying, that there are certain stresses that can be transmitted, that the adaptive response to them can be transmitted through generations.
So I've done a lot of informatics to identify genes that are going up and down responses, and also ones that
yeah. Well, the yeast obviously is a very good model. I mean, the big advantage of the fly compared to C. elegans is that it has a brain.
Right.
And it has sophisticated behaviors that you can measure and assess. And the advantage compared to higher animals is that you don't need permission from the, you can do with them whatever you want. And this is a big thing in the TBI model, because basically all the TBI literature in invertebrates, mammals certainly, doesn't have unanesthetized controls. So everything that you read about, I mean, I don't know whether anybody is interested in neural injury as such, but all the experiments that you see where they test an intervention is always done on the background of anesthetized experimental group and anesthetized control group. And it's just being wiped under the table that actually you're dealing with a brain that has been exposed to anesthetics. So it's not the response of the native brain. And the good thing with flies is that we can actually compare. This is why we were those who can clearly show that it preconditions in TBI. Otherwise, it's very, very difficult to show in mammals, because you have to anesthetize before you inflict a severe injury to a vertebrate, you have to anesthetize. And then you kind of lose the control if you're looking for the effect of anesthetics, right? You don't lose the control for other things, but not for anesthetics, you do lose it. Yes?
Can you talk more about what happens in that 24-hour window for the ND23 mutants between anesthesia and death?
Yes.
Do the neurons fire more or less, or do they die? And is it?
Yes, that's a good question.
To that critical window, or do you see even more and more fly and die 48 hours later?
Yes. So first question. So we see Zach has done these time series experiments watching what happens after you expose the mutant. So the first important thing is both after TBI and after these mitochondrial mutants, everybody wakes up. for flies, it means that they start moving. So nobody gets killed because we were worried that maybe we're just killing them with an overdose of anesthetic. That's not the case. They all wake up. And then at about 2 to 4 hours, we start seeing the first deaths. And that reaches a plateau at about probably 22 to 24 hours. The question, and this answers also your next question, at 48 hours, we don't see a significant increase of mortality beyond natural attrition. Don't forget, these animals are not healthy. So you have a certain natural attrition, but we don't have basically an identifiable increase in mortality beyond 24 hours from this single exposure. So what happens in those, that's the one, that's what the grant, that's what the grant is paying us to find out. So, and we're looking for it. We don't really know yet. We have a couple of kind of working hypotheses, but we don't really know what is going to pan out. Oh, here you are. One more question.
Yeah, I have one more question about ND23 that you've been looking at this in, and then you're also examining it in ND2. And ND23 is this matrix protein, where ND2 is a transmembrane protein full of transporters and stuff. Is there a difference in like the phenotypes of these?
Well, we would like to say, well, first of all, the toxicity phenotype exists. And if there is a difference, it's more qualitative, no, more quantitative than qualitative, with one exception, and that is that sevoflurane, where we have a hard time eliciting a toxicity phenotype in ND23, is definitely toxic in ND2. So this is the one qualitative difference. Otherwise, there are quantitative differences. We have the impression, I mean, the ND2s are sicker, so the mortality rates and also the natural attrition is higher. So it's, but in essence, we reproduce the toxicity phenotype in the ND2s. And we did it, as I said, as a proof of concept. And the second is because we plan to put this mutation into other genetic backgrounds and see whether there are modifiers, modulators of this phenotype in, you know, the flies that we have access to. Okay, well, you think you want to shut down?
That's a good place to end. So let's thank Misha again for one.
Thank you. Thank you so much for, I forgot to do the regular thing, and thank you. Great audience. It was great to talk to you guys.